fly through DNA synthesis, protein synthesis, and glycolysis. You need to look it up and know what it is. Alright, so that's for lecture. What else in lecture test is super important, and that is genetic control of enzymes. This is in our notes, it's in chapter 5, 7 notes. In your book, it's chapter 7. And it has to do with the lac operon, a series of genes working together to control the, for a specific purpose, is called an operon. And operons are named by what they're trying to control. What do you think the lac operon is trying to control? Lactose digestion. And the TRP operon. If you don't read those before I lecture on them in class, you will kill yourself. They're not really hard, but when you first read them, you're going to go, I don't get that, it's too complicated. Ah! Don't kill yourself. Don't even go back over and read it again. I'll explain it when you get here. But remember, it's a two-step process, learning these. The first one is, and the next one is, so they have to be separated by time. So if you don't ever look at it before you get in here, you're going to be, after I go over it, and then where are you going to be when you go, because there won't be anybody to re-explain it to you. So you have to get the at home. Then when you come in, you'll get, I get it. Because, and I know this from experience about me. When I first went over it, I didn't read it before I came to class, so I went. And on the test, I was still at. And for 2 years after it, whenever anybody went over it, I went. And then, when you have to teach it the first time, you have to know it. And that's when it went. So, let me get something to drink.
It's going to take us that long to figure it out. Figure it out.
Now you see what I'm talking about, Tony? Yeah, go look at the first 100 questions. It's all shit.
A sweet person bought me coffee for today, but if I have my 6th cup this morning, I'll be not a nice person. So I'm going to save this for after lunch. All right. All right, so that's on lecture. If I was studying, and if you've looked at the old exam,
yes,
you'll know that there is a tremendous number of 2 kinds of questions. One,
yes,
the cards. And the other?
Yeah, they are.
Genetic control of enzymes. That's where most of the questions come from. What's the third area that all the questions come from? Moving DNA. Which is chapter 8, and we've already been over it a little bit. Conjugation. Translocation. Transformation. Transduction. Those are the, so if you knew this, and finally the cards, you would make 100% plus. Nothing else would matter. Now this was probably going to be the hardest, followed by this. The rest of these you should have had in high school. If you didn't have them in high school, you had a bad high school.
It was like 20-some years ago.
Biology. It's physiology, it's in anatomy, it's in physiology, it's in every biology course. It is the three most universal, oldest biochemical reactions of life. You don't know them, you've got to know them if you know anything about biology. It is the beginning. Genetic control of enzymes is really hard, but if you look at it at home, and you have to start with this one, because this one won't make any sense unless you understand that one. And these are really light, they won't take too much. And if you look at the chapter 8 notes, not the book is a nightmare. The book has pages and pages of excruciating detail that I'm not interested in. I just want you to know these main means of moving DNA horizontally. That means having sex with somebody next to you. Changing genes with them without touching them. Alright, so that's lecture questions. Now let's talk about word questions. Shortcuts to the word questions. Things you need to know about the word questions. It's actually simple. I know. What you need to know is on this. So let's make a list. If it's gram negative, what you need to look for. You need to look for rod or cocci. You need to look for glucose. You need to look for citrate, hydrogen sulfide, indole, lactose, and mannitol. Plus MR and BP. Sometimes motility. That's it. Now there's only a limited number of things that will tell you that. Maybe I better put one more in there, although it's oxidase. So these are gram negatives. We're going to talk about them in just a second, how you get those. But let's look at gram positives. That's why when a bunch of you, I found out I gave you a gram positive unknown, I said, well, shit. That's so easy. You're a smarter person than that. You need more challenge. You need to be able to think more than 3 times, because that's all it takes on a gram positive. It takes that many decisions on a gram negative. On a gram positive, all it takes is shape, catalase, the 3 stains, oxygen usage, nitrate, and casein. Those are easy. So, listen, because a lot of you don't listen. How do I know? I put it on the board. You did 51 cards. And you come up and ask me, why do I have pink and yellow and black in my clear as iron? Why did you do the cards? It says on there exactly why. And yet I have to tell people over and over. In one lab, I had to tell people 21 times, individually, why they had yellow, red, and black in their clear as iron. Those are the 3 colors in the clear as iron. You should know that. It's not hard. Glucose is in the butt, lactose is in the slant, and black is anywhere. Black means hydrogen sulfide, yellow means positive for a sugar, and red means negative for a sugar. That's it. Not hard to learn. Alright, so let's go over this. Shape. If I say longer than wide, slightly longer than wide, slightly wider than long. If I say square, if I say pencil-like a broken pencil lead, any of those things, it's a dang rod. If I say absolutely globular or round, it's a coxy. Anything else is a rod. Coccobacillus is a rod. What does it mean? Roundish rod. Bacillus means rod. So what's cocco mean? Roundish. Roundish rod. Yet people were writing down coccobacillus as coxy. Remember, the first part of a word modifies the second. So any modification of the word rod means rod. So shape should not be a problem. Alright, so that's taken care of. Catalase. There's only two ways to do the catalase test. You put a drop of your bacteria on, a little tiny bit of the bacteria on a slide, and you add 1 drop of catalase hydrogen peroxide, H2O2, and you should see little bubbles. If you don't see them on a slide, you can look under 4x in your microscope. They might be too small to see. You can also put a couple of drops of hydrogen peroxide down an old slant. What's an old slant? One that's at least 10 days old since you made it, and you've scooped all the bacteria off of it or whatever. So any test that uses the word old slant, there are 2 tests that you use the old slant on: oxidase and catalase. This one is hydrogen peroxide, and the positive is bubbles or fizzing. This one has to go from gray to pink to gray to black to be positive. If it goes gray to black, it's negative. And the only time it is ever asked is if you're glucose negative on a gram negative. So when I say the 2 old slant tests, you know I mean these 2. They're the only 2 done on an old slant. If you see bubbles or fizzing, it's probably going to be catalase. Alright, let's look at, everybody should know the 3 stains. The one where you find calcium ions is endospore positive. Or you find holes in rods. If you have purple, pink purple, or hot pink, fuchsia, cerise, rods, plus blue ones,
What is it called?
all the same color in English. It's positive for acid fast, which means it is what genus? Mycobacterium, or it could be Nocardia, remember. But we don't use Nocardia on our sheet. What else can be there? Lipids in the outer wall. If it says lipids detected in the outer wall, all of that is word information that tells you it's a positive acid fast stain. What's all of the things that tell you that it's a positive endospore stain? Calcium ions were detected, holes were seen in the rods, there are some green ovals. casein. But remember, you always got to have a rod. It cannot be inoxide. This one must always be a rod. Gram stain, pink is negative, and purple or purple blue is positive. Pink is negative.
How about only blue?
Still, which is it near?
Purple.
Right. All right, nitrate. Really confusing. If you have reagents A plus B and it's red, it's positive. If you have A plus B and a bubble, then it's positive. If you have A plus B plus zinc and it's red, it's negative. So remember, red can be both positive and negative depending on what reagents were used. If it's A plus B in red, it's positive. If it's A plus B plus zinc in red, it's negative. If it's A plus B in a bubble, actually A plus B plus zinc in a bubble, it's positive. Casein, there are only two. Litmus milk and that means you've got to have whey. What's whey? Clear liquid. Plus a curd. That equals casein positive. So if I say there was whey, you know it's positive. If I say there's a curd, you know it's positive. Litmus, the other casein is skim milk. How do I disguise that? I say the white in milk. Still casein. And if I say the white was removed, it's positive. If I say it was the white remained, it's negative. Remember that litmus milk has another test in it. There are two tests in it. The other one is lactose fermentation. And positive is pink or white. On litmus milk, if I say it remained cloudy, what does that mean?
Negative.
Cloudy?
It's positive.
It
Negative for
means
lactose.
negative for casein.
What?
If it goes clear, it's positive. If it's cloudy, it's milky. So it's negative for casein. Okay, so that's everything you need to know about a gram positive. Not much. Question?
The
ahead.
oxygen use.
Oh, I didn't talk about oxygen use. Sorry. All right. It's wherever there is cloudy. So I say the tube that turns cloudy to detect gas. That would be thioglycolate. And wherever the cloudy is tells you it's oxygen use. So let's look at it. If it's cloudy at the top, it's aerobic. If it's cloudy at the top, but there's clear above and below, it's microaerophilic. Still aerobic. If it's cloudy all the way through, it's facultative. All the way through, but not equally. Notice it's heavy here, heavy there, but thin in the middle. But it's still cloudy all the way through. What if it's cloudy equally? That's an anaerobe called aerotolerant. What if it's cloudy only at the bottom? That's an anaerobe. What else do I use for anaerobes? Carbon dioxide incubator or a candle jar or under oil. Sometimes we put mineral oil on top of a slant. So an anaerobe will grow. So that's oxygen usage. And on aerobes, it'll ask you if it's, on gram-positives, it'll ask you, is it aerobic or anaerobic? So what are the two anaerobes?
Anaerobes are the aerotolerant.
That's right. Obligate anaerobe here and aerotolerant are both anaerobes. What's the aerobes? Microaerophilic and obligate aerobic. What does everything? Facultative. It's bi. Remember what Mom said. If I talk about poop or sex, you'll remember it. Anyone have any questions about gram positives? Remember, on the stains, this is always going to be gram positive, so I don't have to tell you if I tell you the stain. All right, so let's learn one final thing about gram positives. How would you find anything about gram positive if we were talking about a plate? It's gram positive if it doesn't grow on these plates. Heptoin, EMB, and McConkey's. How do I disguise these plates? This one has orange auger. And the positive is hot pink. Hot pink growth on orange auger, or hot pink on hot pink, is positive for lactose. It means it's a gram negative. What is EMB? Purple auger, or burgundy. Sorry. What's a positive? Purple on purple. Black on purple. Because some people say dark purple looks like black. Not black like in the hydrogen sulfide. Or green on purple. Remember, metallic green is heavy on purple auger. I rarely will say EMB. I'll say the purple auger showed black growth. The orange auger had hot pink growth. That tells you the lactose, and it tells you the gram stain. Lactose positive and gram negative. Heptoin, salmon equal lactose positive, gram negative. Black crystals equal hydrogen sulfide positive, gram negative. And finally, green colonies or blue-green colonies on green agar. Salmonella and Shigella. So I use the plates a lot because they'll tell you the gram stain and they'll tell you lactose, and one of them will even tell you hydrogen sulfide. Of course, if I say it was, let's see, the growth was salmon colored with black crystals, on hectoin, you would know it's hydrogen sulfide positive, lactose positive, and gram negative, and a rod. Because gram negative cocci don't grow on any of these. What else? What if it's a salt fermented agar? That means the mannitol agar plate. Don't mix up mannitol agar broth with mannitol agar plate. Mannitol agar broth just means the carbohydrate mannitol was fermented. Mannitol agar plate will not grow gram negatives, only gram positives. What else? Can't think of anything else about gram positives that would amuse me. All right, shall we go to gram negatives? Of course, this applies to gram negatives too, because gram positives won't grow on it. All right, so what's oxidase? We've already been over it. Somebody tell me what word way would I express an oxidase test?
Spacing?
No, oxidase test. An old slant, and what does it have to be positive?
Cloudy.
Nope. Gray, pink, black. Has to have the, remember, positive is almost always pink when it comes to drops. Anything where you're adding drops, pink is the positive, except for one. What's that one? Methyl red. Is pink is positive. Which one you hold it? Is pink not positive with drops? Oh, phenylalanine. Olive green when you add drops. Positive. All right, so we know this one. We've gotten rid of this one. What are the only two tests where you can find your glucose result? Bligger's iron and phenyl red glucose. Or, if I disguise it by saying the three major broths. What are the three major broths? Glucose, lactose, and mannitol. They're always in that order. So if I say the three major broths, two of them were yellow, one was pink. So you're going to have to look for some other test to tell you which one was yellow. Because the yellow is what we care about. So, if I say two of them were yellow and one was pink, what other test would tell you which one's yellow? Okay, if it's KIA in the butt. Down here, yellow means the glucose test. Up here, the slant part, whew,